Now, we've already had genetics, and today what we will be doing is looking how genetics influences the development of a full organism. First, we will look at animal development, and then we will look at the model of human development. Now, when you think about it, how can two cells, a sperm and an egg, having the appropriate genetic material, come together, and suddenly these two cells merge, and you see an organized process of cell division, development, and the formation of a complex organism. There's no question this is a rather complicated process we often refer to as embryology. So when you look at, first of all, we will look at animal development, because animal development has an overall common process. To start with, remember, sperm and egg combine to form a zygote. The zygote forms an embryo, and embryo goes through a developmental process to form a fetus. Now, overall, this process of development, this embryonic development process, we often say it's a self-assembly process under genetic control. Now, cells develop normally from a general to specific functions. General functions lead to a very specific function, like the function of a nerve cell. Now, as we said before, there's a common process to all animal development, of course, which also carries over to humans. If we look at the embryonic development process to start with, there are three parts to it: cleavage, gastrulation, and organogenesis. If you look at embryonic development across species, and here we're looking at snakes, chickens, possums, cats, bats, and humans, and look at how similar the anatomical features cross from left to right, which of course points to a common process. The first part of the embryonic development is cleavage. After the zygote is formed, the new cell starts to divide. We call this cleavage. It will start to divide very, very rapidly to form the morula, which is a package of cells, and then it will continue to divide to form a package of cells called the blastula, which will have a hollow filled package of cells. Now, if you look at what's going on at the blastula state, we can already see that this little embryo has a top and a bottom, the animal pole and the vegetal pole. So therefore, the cells themselves know where the head is going to be and where the tail is going to be. Here we're looking at a package of cells called the morula. Now, you notice that the cells on the top are smaller and the cells towards the bottom are bigger. These cells talk to each other, and they already are positioning themselves to form the various structures of an organism. Now we go to the second part of embryonic development, called gastrulation. Now, in gastrulation, what happens is, if we look at it from the blastula state, where we're looking at here to your left, you see we have the animal pole and the vegetal pole with a hollow mass inside full of fluid. At this stage, what is happening is that cells start to migrate, and this migration of cells forms three major structures: ectoderm, endoderm, and a mesoderm. So gastrulation has the purpose of forming three germ layers Each of these germ layers will form specific tissue and organs, as we'll see in the next stage. As you can see in this table, the endoderm germ layer is going to form lungs, liver, pancreas, and digestive tract, and so on. The mesoderm is destined to form skeletal muscles, skeletal systems, muscles, and so on. And the ectoderm functions to form the entire nervous system and other structures. So let's look at how this process works in organogenesis, which is the formation of organs. Again, the formation of organs is determined by the ectoderm, mesoderm, and the endoderm. Now, the example I'm showing you here is the formation of the nervous system, which arises from the ectoderm. If you look carefully to the right, you'll see that we have three tissues: ectoderm, mesoderm, and endoderm, and we're going to be looking at how the ectoderm evolves into certain neurological structures. This is essentially the same material, and we're looking at a frog embryo development. On the outside, we have the ectoderm, and the ectoderm will slough off cells that will migrate and seed themselves into various positions. At a given position, certain tissue and organs will develop. For example, the neural tube, the epidermis, the notochord, and so on. So this is the overall process as to how given organs evolve during the process of gastrulation, all under the influence of specific germ cells, again, the ectoderm, mesoderm, and the endoderm. So how is all of this orchestrated? The control of development, how does this work? First of all, positioning of cells is important. Cells, after the blastula formation, will find themselves in certain areas. Depending on where they are, will dictate what structures they will evolve into. Secondly, there are a number of proteins called morphogens. Now, these morphogens are produced sometimes by support cells that surround the embryo itself, and even the egg itself, and these morphogens will activate certain genes. Now, this will influence what organs are to be produced in a given area. In addition, cells talk to each other. One cell will direct the development of other cells in a given area, and we call this induction. And another process in this control is a process called apoptosis. Now, apoptosis, we've discussed before, is a process where cells will commit suicide. It's a programmed cell death, so that as an organ is being developed, sometimes there is significant sculpturing that must take place for that organ to fully form and maintain its designated shape. First of all, let's look at morphogens. Now, in this particular process, specific proteins are being produced, and these proteins will influence what is to happen during the embryonic development state. If we look at this figure, you'll see that this particular egg is surrounded by cells, support cells. These cells will be producing specialized proteins. As you can see towards the bottom of the figure, these proteins that are produced by these support cells will migrate throughout the egg itself. As you can see, the degree of, as you can see, the amount of morphogens are more to the left and less to the right. This will signal to that egg after it is fertilized that the head is going to be to the left, the thorax is to the middle, and the future abdomen is going to be to the right. So you can see that very early in the game, even almost prior to fertilization, the egg itself is already being pre-positioned as to where given structures are to evolve. Now, another process that controls embryonic development is induction. Now, we're looking at a chick embryo in the development process. As you can see, this little bulge to your left, the ZPA, this is the zone of polarization, and this little area contains cells that will determine the production of a wing, or I should say, that will determine the development of a wing. Now, if I took this area of cells and implanted it into another chick, so therefore you have two ZPA zones, guess what happens? You get two wings. Kentucky Fried Chicken will be happy with this. You get two wings. And therefore it tells us that packages of cells will determine what is to develop in a given area, of course, during the process of development. So we've discussed a number of processes that control development. Apoptosis, as we said before, is programmed cell death. Here we're looking at an appendage. This could be a human hand that early in the game is webbed. How does this webbed appendage eventually form the hand that we are very familiar with? What happens is that the cells between the digits are sculptured out. They're sculptured out through a process that we refer to as apoptosis, in that the cells themselves know that they are to die, and they are basically eliminated, forming the mature hands and fingers that we see in the fully developed fetus. So, with all of this, all of us have heard of the concept of stem cells. Now, I didn't mention stem cells until now, but really, what are stem cells? When you think about it, early in the embryonic development, cells have the potential to develop into virtually anything. If you remember, in the morula state, we have a little package of cells, and those cells suddenly move into one direction, another direction, and they evolve into various tissues we refer to as ectoderm, mesoderm, endoderm, and these tissues can therefore form various organs, and so on and so on and so on. Now, first of all, early in the embryonic stage, cells are often referred to as being pluripotent, which means that they can give rise to virtually any potential cell. Thus, they are uncommitted and referred to as stem cells. So in essence, the cells in the morula are all basically stem cells. After all, every cell has all the genetic material that every other cell has, which means that every cell has the potential of being anything. But this process, of course, is organized and controlled as a given cell determines what it will be. Now, somatic cells are committed cells. Somatic cells mean cells of the body. A nerve cell is a somatic cell. It is committed to be a nerve cell. A muscle cell is a somatic cell committed to do what a nerve cell does, and so on and so on and so on. So somatic cells are committed cells versus stem cells they are uncommitted. They still are trying to decide what they will be when they grow up. Now, so now we're looking at the potential for stem cells. But the potential for stem cells in therapy. When we think about it, if we have, for example, a heart attack, cells are destroyed, muscle cells are destroyed. These muscle cells cannot, if you remember, muscle cells do not divide. These muscle cells cannot be replaced. Therefore, if we have a cell that we can inject into the heart, a stem cell that we can titillate to become a muscle cell, then the problem is fixed. So this is the potential of stem cells, basically to replace cells that can no longer divide. Therefore, if we have a problem with nerve tissue, we can replace it with potentially stem cells. Muscle tissue, we can replace it with stem cells. Any tissue that cannot heal itself to maturity, we should be able to fix that problem by injecting cells that can be induced to form the tissue that we want. Now, of course, all of us have heard of the controversy involved with stem cells. We have to go to embryonic stem cells, which are usually the best cells to use. However, since all the information for forming a given cell is present in every cell, let me backtrack. Since every cell has all the information necessary to form any potential cell, therefore, even adult cells might be triggered to regress and become stem cells. So much work is being done trying to take adult cells and trigger them to become stem cells. Now here we're looking at embryonic stem cells, just to kind of tie things together here. After fertilization, we see cleavage occurring. Every one of those cells are potential stem cells. If we look at day 5, some of the best at day 5 after fertilization in the blastocyst, some of the inner mass of cells, each of those cells could be pulled out to form a completely new embryo, or each cell could be pulled out to form a designated tissue. So here we see the potential promise of stem cells. Okay, so now let's summarize stem cells. There's significant potential for stem cells in therapy, and you can almost use your imagination as to where stem cells can be used, from Alzheimer's disease to severance of spinal cord. Secondly, the future direction of stem cells probably will be in reprogramming a cell to do what it doesn't normally do. For example, to go to a muscle cell and get it to divide. That would be basically reprogramming a cell. And now, long term, this is going to be a possibility, and this area all of us should really keep track of.
Kept us awake with false teeth, sharing different hearts.